Okay. All right. Well, thank you everyone for coming. So first, I'm going to just do a few of our standard housekeeping items for regulars. You're probably familiar with this. So welcome to Innovative Discovery Series. For CME credit, please remember to sign in and include your email and credentials. Our next talk will be on September 6th, our tech talk, and that will be with Wei Kwan. Am I pronouncing that right? From the University of Delaware. And also on June 15th, our next Innovative Discovery Series talk will have a special guest. Our own Vishal Patel from Christiana Care will be speaking on diabetes and consumerism. So if you can register for both, that would really help us prepare for those talks. And the full schedule of all of the talks and past talks are posted on the CTR website, and that's delaware-ctr.org. So it's my sincere pleasure to introduce today's distinguished speaker, Dr. Ronnie Drapkin, who will be presenting his groundbreaking research in ovarian cancer. Dr. Drapkin received his BA from Brandeis University, and he went on to obtain his medical degree and PhD in biochemistry from Rutgers University. He completed his residency and fellowship training at Brigham and Women's Hospital, and was on faculty at Harvard Medical School and the Dana-Farber Cancer Center Institute before joining the University of Pennsylvania in 2015, where he now holds the Franklin Payne Chair of Gynecologic Oncology. Dr. Drapkin is a distinguished alumnus of the Rutgers University Graduate School of Biomedical Sciences and is an elected member of the American Society of Clinical Investigation. He serves on the editorial board of Cancer Research, Clinical Cancer Research, Gynecologic Oncology, and has authored over 120 articles in scholarly journals. Dr. Drapkin is also director now of the Penn Ovarian Cancer Research Center in the Perelman School of Medicine at the University of Pennsylvania. He's associate professor of pathology in obstetrics and gynecology and the director of gynecologic cancer research for the Brasser Center for BRCA and Penn's Abramson Cancer Center. I wanted to ask you a question. So solve a debate for me. Is it BRCA, BRCA? Do we have a preference?
I think BRCA, either.
Yeah.
BRCA, I hear it all.
I hear it all too.
Yeah.
I was curious.
I think on the research side, you hear BRCA more.
Yeah.
But it kind of all over.
We'll stick with BRCA for today. So within the Drapkin lab at Penn, Dr. Drapkin directs a team of researchers focused on developing comprehensive understanding of genetic, molecular, and physiological factors that drive the development of cancer, with a special focus on gynecologic malignancy. As part of this work, his group has implicated the fallopian tube as a likely point of origin for a majority of advanced ovarian cancers, a finding that has created a paradigm shift in the field, refocusing efforts of experts who long believed the deadly cancers originated in the ovaries. So with that, I'll leave it to Dr. Drapkin to really discuss his pioneering work in zeroing in on ovarian cancer. Thank you.
Thank you, Kimberly, for that wonderful introduction and for the invitation to visit you today. My first time down here, and it's quite an impressive campus. As you were telling me about your CRT and the series, it's hopefully an opportunity for some collaborations. I look forward to that. So the requisite disclosure slide, unfortunately, I don't have anything that interesting that is making me rich, filthy rich. So essentially, it's just some advisory boards I sit on, some funding we get from mostly the NIH. I will be talking about some drugs that are in development, and I'll tell you those stories later. None of those are related to any of the positions I hold at those companies. So why do we study ovarian cancer? If you look at the latest statistics from the World Health Organization, annually there are about 250,000 new cases of ovarian cancer diagnosed annually. Unfortunately, the vast majority of those women lose their battle with that disease within that first year, so 152,000 deaths annually. And that's because most of them get diagnosed at advanced clinical stage. So if you look at mouse here, if you can see my pointer, the vast majority get diagnosed at stage 3 and 4. Once the tumor has expanded well beyond the confines of the ovary, this is an example I had when I was a resident at the Brigham. The surgeon was able to actually take this out all in block, and here's the uterus, just to give you an example how big these tumors can get relative to the uterus. And these tumors will spread and shed, involve the peritoneal cavity, and widespread disease usually ensues. As a result of this clinical scenario, our understanding of the molecular pathogenesis has been limited, and as a result of that, ovarian cancer still remains the most lethal gynecologic cancer in developed countries. Now, another way of looking at these numbers is to look at the lethality index, so the number of deaths to cases that occur every year. And if you look at ovarian cancer, it falls right behind pancreatic and lung cancers with an index of 0.67. And so even though ovarian cancer by numbers is about 1/10 as common as breast cancer, it's about 3 to 4 times more lethal than breast cancer. Now, adding to that complexity is the fact that ovarian cancer is heterogeneous. There are many subtypes of ovarian cancer. It's not just a single disease, the most common of which is the serous carcinoma. And that's what I'll be focusing most of the talk on today, because it's not only the most common, it's also the most difficult to treat, and we still don't have any curative therapies for it, nor early detection methods to detect it at a stage where we can make an impact on overall survival. So the talk I'm going to break into 3 parts. I'm going to tell you a little bit about our understandings of the origins and the genetics of this cancer and what the clinical implications are, because we're actually seeing already clinical implications based on this new. And then I'm going to sort of drill down a little bit into this concept of copy number alterations. And I say copy number alterations because I contrast that with mutations. We tend to think of cancers as associated with lots of mutations, but changes in the number of genes amplified or deleted, copy number changes, are actually a major manifestation that are the hallmarks of this particular cancer compared to other cancers. And then ultimately, at the end of the day, what do we do about all this? How do we translate this into the clinic? And I'm going to give you a couple of examples of things that we're excited about as we move forward. So let's talk about the origins and genetics of ovarian cancer. Rewind, about 20, 25 years now, the BRCA genes, the BRCA genes, whatever we want to call it, were cloned in 1994, 1995. And by the end of that decade, we were offering women who carried these mutations prophylactic surgery to reduce the tissues at risk, which meant prophylactic mastectomy for breast cancer and prophylactic removal of your fallopian tubes and ovaries to reduce the chances of getting ovarian cancer. And the idea was that if you took these tissues out early enough, not only would you spare women the morbidity and mortality associated with getting this cancer, but we might have an opportunity, especially as pathologists, to look at these tissues and see, can we find out where these early cancers actually begin, thinking that it was somewhere in the ovary. And so what happened over the ensuing years was that people studied these specimens extensively, always looking thoroughly at the ovary, never, unfortunately, ever finding any consistent precursors that we could put our finger on. But what we did have was anecdotal reports as early as 2001 showing that in some cases there were always these funny-looking cells in the fallopian tube. Now, just to give you an idea of how we used to process these specimens, right? The ovary would come out, the fallopian tube would come out. So my arm is like the fallopian tube, and this is the fimbriated end. The ovary would get sectioned and submitted for histologic evaluation in its entirety. The fallopian tube would get a section through the middle. We'd look to see if there was anything there, not really expecting to see much there, because if you looked at incidence of primary tubal cancer by WHO criteria, it's exceedingly rare. And then literally the rest was tossed, right? It would stay in the bucket, not examined. So the interpretation of these anecdotal reports at the time was that, so if you have a BRCA mutation, perhaps you're at risk of getting breast cancer, ovarian cancer, and fallopian tube cancer, right? Now, around 2005, 2006, I was still finishing up my training at Brigham. I was lucky to be working with Christopher Crum, who's the head of our GYN pathology department. And he said, you know what, this is just too strange. We have all these anecdotal reports of weird cells in the tube. We've never found anything in the ovary, but no one has ever systematically studied the tube. So he said, well, we should do a really thorough characterization of what's going on in the fallopian tube of these women who have these BRCA mutations. And so he developed what we call the SEE-FIM protocol. SEE-FIM stands for sectioning and extensively examining the fimbriated end of the fallopian tube. And the whole point of this was exactly that, just to examine all the epithelium we could, focusing a lot on the fimbriated end, sort of more distal part of the fallopian tube that really kind of hovers over the ovary, and cutting it into enough sections to expose as much surface area as possible so we could see what's actually going on. And that was really when things started to change, because from that point on and over the next 5 to 10 years, what we found and what others have really corroborated in many studies throughout the world is that the vast majority of these serous cancers, the high-grade serous cancers, that most common subtype of ovarian cancer, starts in the fallopian tube. And here's an example of what we think is going on. So normal fallopian tube has ciliated secretory cells. Maybe you can make out the little ciliated projections in those cells. And if you stain for P53, which is the most commonly mutated tumor suppressor in all of human cancer, but particularly in ovarian, you see that there's really little staining. We're looking for brown staining. And that's because the normal wild type P53 gets turned over every 20 minutes or so. The only time you ever see staining of P53 is if it's activated because of DNA damage. And then we found in these cases that we had these stretches of benign-looking secretory cells that stained very strongly for P53. And that was kind of odd because they looked benign, they didn't proliferate, but they stained very strongly for P53. And occasionally, and this is rarely, about 5% of the time or less, we would find these in continuity with what looked like an intraepithelial carcinoma. We called those tubal intraepithelial carcinomas or TICs. Now you hear a lot about STICs because we say serous tubal intraepithelial carcinoma. And those would then be in continuity with that clinically evident disease, the invasive carcinoma. And so those are sort of the histologic observations that were initially appreciated in mid-2000, 2005, 2006, again, reproduced over the years. A lot of molecular studies have been layered onto this. We, for instance, now know that these benign-looking cells that stain very strongly for P53 actually do have a mutation in the P53 tumor suppressor. And most of what we appreciate about ovarian cancer, which is the clinically evident disease, came from the Cancer Genome Atlas, the NIH's effort to really understand human cancers as in-depth as possible. So the goal of the TCGA, for those who aren't familiar with it, was to collect approximately 500 cases of any given tumor. The first three that people looked at were brain tumors, lung cancers, and ovarian cancers, because we have no early detection for either of those, and we have no curative therapy. Right? The 5-year survival rates for all of them are terrible. And so ovarian was one of the first that got analyzed in this way. This was not done at any one place. It was done across many centers in the country. A lot of bioinformatics, really cold, all of these different approaches. And what we learned from them is the following. This is a somewhat complicated slide, but I'll walk you through it. In the top panel A, you're looking at what we refer to as copy number alteration. So from chromosome 1 all the way down to 22. Across a variety of different tumors, so bladder cancers, breast cancers, colorectal, GBM, head and neck, and so on. Here's ovary. And what you're looking at is red, signifying amplifications of different parts of the genome, and blue, signifying deletion. And really, from a 50,000-foot perspective, what you can appreciate is that ovarian cancer is the most complicated genomic terrain that's ever been described. Every single chromosome is affected by amplifications or deletions. And yet, when you look at panel B, and you look at the actual somatic mutations that are aligned with each of these different tumors, you find that ovarian cancer actually has a relative paucity of somatic mutations. So really, it's a copy number disease. Somehow, the genome goes from being diploid and normal to completely scrambled across its entire genome. And we were actually quite interested to figure out, how does this happen? How do you go from cells that look perfectly benign to a genome that is completely scrambled in every which way that we can identify? And so about 6 months ago, we published a study where we went sort of back in time. Now that we know where to look, now that we know the precursors exist in the fallopian tube, we said, could we go back in time, identify those precursors all the way down to that normal cell, and use whole exome sequencing and copy number analyses to figure out how and when does this occur? And so this was a great collaboration with a colleague of mine at Hopkins, Victor Velkulescu, who's a very well-known cancer geneticist, and a very talented postdoc in my lab, Intidar Leviti-Galli, who actually now has her own lab in Geneva, and a very talented postdoc in his lab in Nico Pat. And what we did was essentially identify cases of advanced ovarian cancer where we could identify all the precursors, the normal epithelium, what we call the P53 signature, the intraepithelial carcinoma, the ovarian tumor, and then various metastases in the peritoneal cavity that might have been present in any one of the patients. We also looked at risk-reducing samples from patients with BRCA mutations, and we tried to sort of limit the analysis to cases that were relatively fresh, meaning the blocks, the tissue blocks, were relatively fresh because we wanted to make sure the integrity of the RNA and the DNA was intact. We then used laser capture microdissection to isolate the epithelial part of all of these lesions, and then did whole exome sequencing and copy number analysis to figure out what's going on. We had the luxury of working with Rachel Karchin at Hopkins, who is a great computational biologist and bioinformatics expert. And she was actually able to use this mutation data to develop a molecular clock that would essentially tell us how much time transpired between the normal epithelium and the acquisition of all these precursors until the time when the tumor evolved and became clinically evident, right? Which we had really not had a sense for before. So let me walk you through some of that data. It's a little bit complicated, lots of colors. Essentially what you're looking at are 5 patients that we looked at. Patient 1, 2, 3, 4, and 5. On the left, you have these early precursors, the signatures. Right next to it is what we call that STIC lesion, or the intraepithelial carcinoma. Next to that is the invasive tumor. And then as you go from left to right, it's the ovarian tumor and then various metastases in the peritoneal cavity, right? So you're going from benign to gradually more malignant as you go from left to right. And what we found was that the first mutation we see in all ovarian cancers is a mutation in P50. So essentially 100% of all high-grade serous ovarian cancers have a P53 mutation, and this is totally consistent with what the Cancer Genome Atlas taught us, right? It's essentially the only cancer that it has an obligatory mutation. Okay? But even in these early precursors, a signature, where we didn't know what else might be going on, there are other things going on. Every patient has their own unique personal or private mutations, and there's also the beginnings of copy number alterations being seen even in these benign appearing, although mutated cells. So there's already something happening at a very early stage with the acquisition of the P53 mutation, as well as copy number alterations. As you progress from this lesion here to the in situ cancer, you acquire additional mutations and additional copy number alterations. The important part is that every single one of these alterations is then passed on to the more malignant progeny, right? So that you retain all those alterations as you move forward. You can see that that P53 mutation is present throughout all the lesions that we studied in any one given patient. Here's just another example where you see the signature here, acquisition of additional mutations in the STIC, and for instance here, if I'm not mistaken, this is an omental mass that had acquired additional changes. Again, here's another example. And so as you progress away from the tube, you acquire more and more alterations in the genome. Sort of in summary of what we learned was that yes, P53 mutations are early, that copy number alterations are also very early, and that was actually quite surprising to us. In fact, loss of heterozygosity of LOH for P53 was thought to be a somewhat later event, actually happened pretty early, which was not expected. And then that molecular clock I told you about, the calculation was that if you go from that initial in situ lesion, that precursor that's an intraepithelial cancer, it takes about 6.5 years for that lesion to get to the ovary. But once it gets to the ovary, it takes less than 2 years for it to spread everywhere else. And so it seems that the window of opportunity is the intervention or the detection of that precursor in the fallopian tube before it ever gets to the ovary. Once it gets to the ovary, you know, it's sort of way out of the barn, right? The horses run free and wild, and it goes all over the place very quickly. And so that was something that we hadn't appreciated before, but clinically it made sense. We also wanted to study the evolution of these tumors. How do we know how they evolve at a genomic level? And so this is, again, work that was done with computational experts using a number of different algorithms, essentially all showing us the same thing. And these are, again, our 5 patients using now our phylogenetic trees to sort of give us a sense for how these tumors keep. And so all of them have a P53 mutation. Again, just reaffirming the fact that it's an early event. And the patterns of evolution are a little bit different. Yes, we have cases where there's a linear pattern of evolution where you go from the fallopian tube to the precursor signature to the in situ carcinoma to the fallopian tube to the ovary, and it just goes in that direction, right? But there are other examples where there's what we call a branching phylogenetic tree. And that reflects the fact that there's ongoing genomic instability even in the precursors. They're continuously undergoing DNA damage and genomic instability and continue to evolve sometimes away from each other as you progress. One very particularly interesting finding we had in this particular patient, and this is the arrow that I'm pointing to here, is that this tumor started on the right side. And on the left side, when the pathologist got the specimen, they said, oh, it looks like there's a STIC lesion, an intraepithelial lesion on the left side as well. And that's not terribly uncommon. We have seen precursors on both tubes. But they called it a stick lesion. When we did the genomics, it turns out that what looked like a stick lesion was actually metastatic droplet that had gone from one side to the other. And genomically, we were able to sort of identify it as such. But it's sort of a cautionary note that in the setting of advanced disease, anything goes. So what you think that might look like a precursor on one side, you got to be careful because it may actually be a metastasis from the other side. And that was just sort of a warning for us. But in hindsight, we know ovarian cancer can go everywhere. And so it wasn't terribly shocking, but I think it's important to sort of keep it in mind. Okay, so then the question becomes, well, are all stick lesions, are all precursors created equally? The reason we ask this question is because we know in the setting of cervical cancer caused by HPV, right, that there's high-grade lesions and low-grade lesions. The low-grade lesions, which are caused by HPV 6 and 11, tend to sort of regress oftentimes on their own, and we don't necessarily need to treat them. We just have to follow them. The high-grade lesions are high-grade. They will almost inevitably invade and become an invasive disease that can create lots of problems. The question is, are stick lesions in the tube segregating in similar ways? Are there bad ones? And are there ones that are not so bad, right? Do we need to necessarily treat every single one depending on the genetics? And so we wanted to start asking the question of, are there precursors for ovarian cancer that might be stratified into different categories? Preliminary, the numbers aren't big here, but one of the things we found was that if we looked at these stick lesions that were associated with invasive disease, right, they had on average about 33 alterations in their genome. In the BRCA mutation population that were having prophylactic surgery, right, so they were normal women, healthy, were having their tubes removed because they had a BRCA mutation. Theoretically, there should be nothing there, but we know that on occasion, 4 or 5% of the time, you will find an early precursor in their tubes. These are those cases. We looked at what the genomics of those lesions looked like, and this gives you an idea of the range of alterations we found there, which kind of falls within the same ball, right? So at least with this very limited look at what we call incidental sticks, it doesn't look like they're any more or less complicated than sticks that are associated with invasive disease. So you could interpret this as saying all sticks are potentially bad, but I would caution that it's only 4 cases. But this study actually led us to apply for one of the NIH Moonshot grants, where now that they've got the TCGA, the Cancer Genome Atlas, what they want to do is exactly what we did here. They want to move back in time and look at the pre-cancers. How did these cancers evolve? And so they actually put out an RFA for pre-cancer atlas. And so we thought we should do this, and we spent our winter break doing this. We should. I think it's actually getting scored today or tomorrow, so we'll find out quite soon. Fingers crossed. Yeah. It's very competitive. We'll see. So where does this take us? What are the take-home messages, right? Well, we know that precursors to high-grade serous ovarian cancer exist in the distal end of the fallopian tube. We actually believe that the majority of these cases actually begin in the tube. That really mutations in P53 are early events, as are copy number alterations. And that the genetic evolution of this disease says that there is a window of opportunity between the onset of a precursor and the development of a clinically evident ovarian cancer. That window of 6 to 7 years is really our opportunity to focus on prevention. And then again, that cautionary note that in advanced disease, a stick lesion may actually be a metastatic event. So, what are the implications of all this for prevention or early detection? The question we always get is, okay, great, so what? What can we do about it? Which I think is a fair question. So I'm going to give you a 1-slide summary of what's actually going on. In terms of prevention, we know that removal of the ovaries in premenopausal women can lead to surgical menopause, essentially what we refer to as contracting that normal 5 to 7 year window when you transition into menopause naturally into an immediate event, right? And that has all its complications with cardiovascular, bone, and other organ dysfunction. So about 30% of women, depending on which population you look at, whether it's ethnic population or geographic population, will refuse to get that surgery because they do not want to deal with the morbidity associated with this operation. So for those women, the Society for GYN Oncology now recommends the following. They said, look, we think that now all these tumors begin in the fallopian tube. You should have what we call integral self-injectomy and delayed oophorectomy, right? Take out your fallopian tube first, and then come back and take out your ovary after you've completed your natural menopause, okay? the hope is that we'll see a similar risk reduction in that population of patients. And there are two ongoing studies now in the country, one called the WISP study and one called the TUBA. These are prospective studies that are actually offering this approach to women at risk.
So I'm sorry,
Yes,
when you say 30-40% of carriers, that's the carriers of the BRCA?
Yeah.
Okay.
The high-risk women, which is the vast majority are BRCA mutation carriers. That's a loaded question in some ways because our screening for high risk is now expanding into multi-gene panels, right? It's not just BRCA1 and BRCA2, it's the RAD51Cs and all these other genes. You know, some of them are not as highly penetrant, some are more moderately penetrant. We don't necessarily know what the implications of all those are, but the technology is there and it's happening. So it is a little broader than just BRCA1 and 2, but that's generally what we refer to. Now, what about women at average risk? Right, so now you don't have a history of ovarian cancer in your family. What is the recommendation there? And again, the American Board of OBGYN says if you're going to have a hysterectomy for any reason, fibroids, very common, right? The surgery was you take out the uterus and you leave the tubes and ovaries because do no harm, do no more than you need to do, right? That was the thinking and the right thinking. Now, based on this new concept of where these tumors start, they're like, take out your tubes, right? Opportunistic self-injection. If you're in there for any reason to take out a uterus, take out the tubes while you're there. That uptake of that recommendation has gone almost up to 75% or 80% of the hysterectomies now in the States. It's unbelievable how that has sort of taken off with regards to practice. Yeah.
So did they find, what's the percentage of early cancer then did they find?
In the normal population, it's exceedingly rare. I think it's it's around 0.2, 0.3%, something like that. It's very uncommon. Right, so it's really that defect in BRCA that predisposes you to that. The risk in the average population is less. That being said, the vast majority of ovarian cancers are sporadic as far as we can tell. So that is also changing as we do more genetic analysis. Yes, please.
What's the morbidity associated with self-injection?
So that's a great question. The concern, right, is could you actually take the fallopian tube without compromising the blood supply to the ovary? Because if you compromise the blood supply, you essentially achieve nothing, right? You're going to put the woman into menopause and you left the ovary, right? So there are a number of studies that were done over the last, now it's probably like 5 to 10 years. Small studies and then larger studies arguing that, hey, it's a very simple surgery to do if you've never had prior surgery, meaning no issues with adhesions or other complications. And so far, the studies that have been done with follow-up, looking at hormone levels and symptoms of menopause, suggest that it's not doing that. So what the actual primary endpoint for the WISP study, which is one of these prospective studies, is quality of life. It's looking at that. The secondary endpoint, which will take more time, is to figure out did we actually risk reduce you as effectively as the standard therapy. So that's already happened, right? These are things that are actually already happening in the clinic as a result of Cydin E1. What about early detection? Some may be familiar with this huge study that was published in the UK, the UK CTOC study. The epidemiologists in the room might be familiar with that. Ian Jacobs and Lucia Menon, where they looked at the combination of CA125 and transvaginal ultrasound. Both methods on their own have not been shown to be effective for early detection. The thought was if you use multimodal screening, so combine two approaches, could that be better? The interim analysis came out about a year and a half ago, suggested that there might be a signal that the goal was overall reducing mortality. Perhaps there's a signal there. The study final report is supposed to come out at the end of this year, and we'll see if there's evidence that that works. I think most people find that it's going to be likely difficult to have just a two biomarker approach that's going to work to detect all ovarian cancers. Liquid biopsies are very popular right now. You've heard a lot about them in lung cancer and melanoma. The question is whether there's a role for liquid biopsy in ovarian cancer. So we're working. This is an opportunity, I think, for collaboration with Christiana, because I think every one of our patients that comes through the door that's going to go even through standard therapy could essentially have a blood draw and follow it over time to see whether we can find things in the blood that predict whether you respond to therapy or recur more readily or faster than others. That hasn't really been done yet. It's an easy one to do, and we want to get a consortium of hospitals that are interested in doing something like that. So that's an area that is evolving. I'll tell you about two others. One is the PAP gene, which is out of Hopkins again. And what the concept there is, it's what we call tissue proximal sample, right? If you know where the tumor begins, go as close to the site of origin as you can, because a lot of the factors that get secreted by the tumors, when they get out to the periphery, are either not abundant enough to be detected above the more prevalent proteins in the blood, or simply don't make it out to the periphery. So go to the source, right? Well, now we know where the source is. It's the fallopian tube. Well, can we get close to the source? So the question is, can we use a pap smear or an endometrial approach to sample fluid that would report on what's happening even higher up in the female reproduction? And so they're using genomics to sample this fluid from the uterine cavity in the cervical region and to see whether they can detect defects that reflect the presence of a cancer in the tube, which is further up. Since flow goes anterograde, the idea is that it would flow in that area, you should be able to catch it. That's very exciting. There are a number of high-profile papers that have come out in the last 6 months that suggest that that may be an opportunity. And then the last one, which I'm really excited about, is what I call the falloposcope. So there's a company out in California that was started by bioengineers, very smart people, and they said, look, why reinvent the wheel? Hysteroscopy is a well-established procedure. It's an office procedure. It's a way to look into a female's uterus when there's questions of infertility, abnormal bleeding, when you want to take a biopsy. It's a well-established, easy procedure to do. If we could modify that hysteroscope so that we could actually cannulate the fallopian tube, right, and sample that epithelium, that may be a way of essentially providing us the equivalent of a pap smear, but of the tube, right, much further up. So they developed this falloposcope, and I was actually out there last year, and they showed us a production facility. They've already done a phase 1 study for safety at the University of Virginia. They had 40 patients, 80 tubes, the math, and there were no adverse effects. They actually found a early precursor in one tube, and these were women that were going to have surgery anyway. So then they were able to compare what they found on their cytology with the pathology that came out, and there was a concordance of greater than 93%. So now they're going to open up a national study. We're going to be part of, Penn's going to be part of their phase 2 study, looking at women who are going to have surgery because they have an abdominal mass. You don't know if it's benign or malignant, and women at high risk, right, which is really the population we ultimately want to have a tool like this for. So that we're excited about, because I think there's going to be an opportunity there to really sort of think about what we could do with these samples genomically, immunohistochemically, and so on to really help figure out what's going on. So that's how the tube has informed clinical practice to date. Copy number alterations. So here's just an example. Another way of looking at the ovarian cancer genome, right? Red is amplified, blue is deleted, and you can appreciate from chromosome 1 all the way to 22 here, it's a mess. Right? Every region is affected. And one of the more common alterations is a gene on chromosome 19 called Cyclin E. Now, Cyclin E amplified tumors are notoriously bad tumors. They're not just bad in ovarian cancer, they're bad across all cancers. They're associated with resistance to standard therapy and poor prognosis. Okay? So what does Cyclin E do? Well, for those who remember the cell cycle, right, we have the G1, S, G2, M, right? S is where DNA gets replicated. And this is important because that's where Cyclin E and its partner CDK2 form a complex that really helps trigger that transition from G1. What happens when you have amplification of Cyclin E is that the levels of cyclin do not cycle. They stay constant throughout the cell cycle. And so that actually causes premature entry into S phase. So G1 gets shorter. You enter S phase before you're ready. That causes replication stress, DNA damage, and ultimately in M, it causes centrosome amplification. Bad things when you think about the development of cancer. Now, we've known this for a very long time, but no one actually understood why is it that Cyclin does this? How does it actually do it? Until a very recent paper in Nature was published, I think, in March of this year by Thanos Halzanidis. This is a great paper where he showed that because Cyclin E forces the cell into S phase early, there are ectopic DNA replications or origins that are normally silenced during G1, but now are exposed and are used for replicating DNA and create conflict between the DNA transcription machinery and the replication machinery. These complexes collide, and you get breaks in the DNA. And so he was able to actually map all the regions in the genome that are affected by this, and at least now provides us with an understanding as to why is it that Cyclin E causes this. It's because now we understand it's causing an ectopic activation of all these normally dormant origins to fire when they shouldn't. Now, is cyclin E amplification unique to ovarian cancer? Kind of alluded to the fact that it's not. Here's just a table that looks at this. So in A, which is kind of small here, and really the point is that along the x-axis here are the names of all the different tumors that have been characterized by the cancer genome map, which is a lot, right? In red are amplifications of cyclin E. Here's high-grade serous ovarian cancer. What you can appreciate is that across a variety of solid tumors, amplifications of cyclin E are not infrequent events. And as I mentioned, in most tumors, this is associated with badness. Okay? Now, in ovarian cancer, which is what we're looking at here in the fallopian tube, which progression I showed you, cyclin E amplifications account for about 20% of these tumors. And they are mutually exclusive with mutations in BRCA. That observation was actually one that was reported in the original Cancer Genome Atlas paper that was published in Nature in 2011, but no one understood why are they mutually exclusive, right? Now we know, and I'm not going to get into the data, but we know that they're mutually exclusive because they're what we call synthetic lethal. A cell cannot tolerate to have a mutation in BRCA1 and an amplification of cyclin E because of the stress it puts during S phase. Too much damage, it's catastrophic damage, and the cell dies. So you never see those two events occur together. If you have an amplification in cyclin E, you will not find a BRCA mutation and vice versa. The other thing we learned is that these amplification events in cyclin E are early events in ovarian cancer. Since now we know where to look in the fallopian tube, we can start to map when does any particular gene alteration occur. Is it early? Is it a late event? And so we found that dysregulation of cyclin E occurs as early as some of these precursors that we described in the fallopian tube, which was surprising, again, because at the time we didn't appreciate the copy number changes that were happening so early on. So we thought, could we model this in the lab, right? Could we model this transformation by cyclin E in the lab, since it seems to be an early event? And we've spent the last 10 years or so developing different model systems to study the fallopian tube, really ultimately designed to figure out what makes it so susceptible to transformation, right, to becoming a cancer. And we've developed primary cell line cultures, immortalized cultures, genetically engineered animal models, and what we call the patient avatar, where we essentially take a patient's tumor, implant it in an animal, and propagate it. And the incredible thing about that is that those tumors retain fidelity to the original patient tumor. Genetically and how they look, they remain constant. Those are great models for drug discovery and drug development. For this purpose, though, we focused on our cell line models, where we can actually immortalize these fallopian tube cells and ask, what's a consequence of overexpressing cyclin E in this setting? And so this is work that a very talented postdoc in the lab, Allison, had done some years ago, where she took these cells from surgery, immortalized them with telomerase and a mutant p53. She put in that mutant p53 because I told you, mutations in p53 are obligatory in this disease. So any model that you start with should have a mutation in p53 because it's going to reflect the human condition. And then she overexpressed cyclin E, or just an anti-vector as a control. And what she found, and I won't spend too much time on this as it's published, is that you could get activation of its partner, CDK2. These cells grew faster, they were more clonogenic, and they were able to grow without sort of contact inhibition. The normal property of normal cells is that once they come in contact with each other, they stop growing. These cells kept on growing and piling up on top of each other. That's what we call loss of contact inhibition. But they couldn't grow under anchorage-independent conditions. They couldn't grow in 3D. Right? Most cells, when they detach from the basement membrane, they die. Right? They're like, uh-oh, right? This is anoecus, and they die. Cancer cells don't, and so they can grow in suspension. These cells could, suggesting to us that cyclin E wasn't quite enough to transform these cells. But they did cause plenty of DNA damage in the cells, and we can measure this by looking at phosphorylation of a particular histone that gets phosphorylated at sites of double-strand breaks, or using comet acids, which is a somewhat cruder tool to look at the extent of DNA damage in these cells. So they were being damaged, but they weren't being totally transformed. The interesting part is when we profile these cells, this is what we refer to as a volcano plot. The important part here is just to focus on the cells in the upper right. The cells that express cyclin E are damaged, right? They go into S phase prematurely, and they upregulate all the factors that protect the replication fork. It's as if in order to tolerate the stress that they're being pushed into, they need to upregulate the DNA repair pathway. BRCA1, all the FANC genes, a lot of the factors involved in stress repair. And so it suggests that there's a upregulation of these, and from other data that people have worked on, also a dependency, because they're all synthetic lethal. Get rid of them in these amplified tumors, These tumors die, they can't tolerate damage. That's why BRCA1 is on that list, right? They're mutually exclusive. Once you upregulate it, you can't take it away. The cell depends on it. And we could verify this by Western blots. We could show that there was upregulation of some of the FANC genes and RAD51 parallel. And so another way of looking at this is half of high-grade serous ovarian cancers are associated with BRCA defects or defects in homologous recombination. And these are tumors that tend to do well when you treat them with DNA-damaging agents because they can't repair the DNA damage. And the other half are what we think are HR proficient, and Cyclin E is the poster child because those tumors don't respond to anything and are associated with poor prognosis. So how do you treat this, right? And this transitions us into what can we do about it? Well, in Cyclin E, we published a paper last year where we looked at a panel of cell lines, ovarian cancer cell lines, up here, that have amplifications in Cyclin E, a gain in the locus, or were unamplified. And what we found is when we knock down Cyclin E or CDK2 in those cells die. They absolutely depend, they're addicted to Cyclin E and CDK2. These other cells could care less, right? If you don't have an amplification of Cyclin E, the cell doesn't care if you take it away. But these cells do, right? And so the question is, can we drug those cells? Can we somehow find drugs that could do the same thing? The obvious partner for Cyclin E is its CDK2 kinase partner, which has been developed and has been tried to be developed for a long time as a therapeutic target. We were particularly intrigued by this possibility because of the recent excitement around other CDK inhibitors like palbociclib for breast cancer, CDK4/6 inhibitor here. And so we went and looked at what's available in the world of CDK1/2 inhibitors and wanted to see whether we could find a drug that would give us the same amplicon-dependent dependency that we saw by knocking down Cyclin E, CDK2 in those lines with the application. So we looked at Dinaciclib, which is a drug that was developed by Merck, and asked, do we see an amplicon-dependency in these cells? And unfortunately, we did not. Right? And part of the problem is that Dinaciclib, it's a CDK1, CDK2, CDK9, probably CDK7, and other CDKs that are important in transcription. And that's always going to give you toxicity. So we don't have a CDK-specific inhibitor, which is a problem. So what can we do? So we did two things. We did two studies that eventually converged. One was done by David Botel's group, and we helped a bit there, where he did a small molecule screen. He said, okay, if Dinaciclib is too toxic by itself, could we reduce the dose and see if we could find another small molecule that would cooperate with Dinaciclib to kill these Cyclin E cells? That was the thing. And then what we did was a gain-of-function screen, because you remember I told you Cyclin E alone was not enough to transform the cells. So we asked what other genes could cooperate with Cyclin E to fully transform the cell. So this was the screen that David's lab did. 4,000 compounds representing all kinds of your typical targets, epigenetic, or receptor tyrosine kinases, and so on. I'll jump to the punchline. What they pulled out was an AKT inhibitor. MK2206 was a compound Merck had developed some time ago. And what they observed down here at the bottom is that there was synergy only in the cells that actually had the Cyclin E amplification. The cells that didn't have the amplification, they did not see any benefit of combining Dinaciclib with an AKT inhibitor. But in the cells that had the amplification, they did see it. Here's sort of another way of looking at this. They then looked and said, how specific is this AKT inhibitor? Excuse me. They got an AKT inhibitor, the one from Merck or GSK. And in both cases, the combination was particularly synergistic in the cells that were amplified for Cyclin E, not for the cells that were not. So this data really drove us to go into animals where we looked at a xenograft model of OVCAR3, which has the amplification, or COV3, which does not. And what you can see is that the combination therapy of Dinaciclib and the AKT inhibitor in the amplified line essentially resulted in no tumor growth, whereas in the unamplified line, there was very little effect in the ability to thwart the growth of these tumors. So I'll tell you that as exciting as that is, it's been very hard to move that into the clinic. Merck has no interest in developing Dinaciclib further, nor their AKT inhibitor further. And so we've been trying to find partners, talking to GSK and others to try to see if we could find a combination that would work. The reason that we wanted to do that was based on the gain-of-function screen we did, which was based on what I showed you before, these cell lines that we had overexpressed Cyclin E in, but did not find that they were sufficient to fully transform the cells. So we asked what other genes could transform them. we didn't do a whole genome screen. We actually limited our screen to just the cells that are amplified in ovarian cancer. We thought that those would be relevant. No point in screening for genes that get deleted. So we developed a library with the Broad Institute of all the amplified genes in ovarian cancer. We introduced this library into our cells that carry the p53 mutation cyclin E and simply asked which genes enable these cells to grow in 3 dimensions, right? Anchorage independent. So we got some hits, and among the top hits we got were AKT. So again, out of the small molecule screen, we got an AKT inhibitor. Out of this gain of function screen, we got AKT. We were able to go back in the lab and introduce either AKT1, 2, or 3 into our cells, and we found that when you introduce AKT2 or AKT3, these cells are now able to grow under anchorage-independent 3D conditions, suggesting that they were transformed. We have subsequently put those into animals, and they do form tumors, so they are transformed. So under 2 scenarios, a small independent molecule screen, you got an AKT inhibitor, and a gain of function screen says AKT cooperative. So again, it's required, and it creates a dependency that might be druggable. We actually went back and asked which AKT is actually important, because there's 3. So we looked at all the members of the pathway and asked with the CBio portal, which is a great platform for looking at TCGA data, which ones correlate statistically. And the only one that correlated statistically was AKT2. So this is sort of just a higher power view of that. So why AKT2? Turns out AKT2 is on the same chromosome as cyclin E, and they oftentimes get co-amplified when you get amplification of this locus. And so they cooperate in the transformation of these tumors, also creating a dependency that we can target with an AKT inhibitor. So that summary of that particular approach led us to identifying this combination. I sort of put a pause button down here because we've struggled trying to move this into the clinic simply because of the difficulty finding a pharmaceutical partner that wants to push this forward. They all love the combination. They all love the data. They have one drug. They don't have the other drug. They don't want to play with this one. It's the usual challenge. And so we're trying to move forward with that. What we're hoping for is that we'll find either an academic or some medicinal chemist who's interested in making CDK-specific inhibitor, because I think if we had that, that would actually really catalyze things forward. But the last piece is a more positive result. We were still interested in trying to understand how a cell tolerates chronic cyclin E, right? And so we did a time course where we could induce cyclin E expression in these fallopian tube cells. And what you're looking at here is just a Western blot where you see over time, hours to days, you get induction of cyclin E expression. And what we noticed was that there was activation or phosphorylation of CHEK1, which is one of these DNA repair pathways that is oftentimes activated in these cancers. And this is where proximity really made a difference, because my colleague Fiona Simpkins, who's a GYN oncologist and a junior faculty member in our center, had been studying BRCA mutated cells and noticed that when she treated BRCA mutated cells with PARP inhibitors, she also saw activation of the ATR CHEK1 pathway. And so she then looked at cells that were resistant to PARP. They already had activated the ATR CHEK1 pathway. Cells that were resistant to platinum also had activated the pathway. So that suggested that maybe ATR and that CHEK1 axis is actually a critical sort of node across tumor types, across ovarian tumor types. And so we went back to our cell lines and asked, could the combination show efficacy in vitro? And what we found is that single agent across all these cell lines, HR deficient, really the BRCA mutated cells, the PARP inhibitor resistant, or the cyclin E amplified cells, didn't really work, but the combination actually gave us really nice effects that were all within the synergistic. So then we said, okay, let's see if we can drive this in vivo. We went back. The Penn Ovarian Cancer Research Center has a really rich platform for these avatars, these patient tumors that grow in mice. And so we've been able to develop these in animals and actually make the cells bioluminescent so we could track disease burden without having to sacrifice the animals. And so we identified and marked animals that had BRCA mutations, that were PARP inhibitor resistant, and that had cyclin E amplifications to ask, could this combination actually prevent tumor growth in this setting? So here's the results with our HR deficient PDX. This is the BRCA mutated ones. The combination gave us complete tumor regression. The PARP inhibitor resistant models also gave us complete tumor regression with the combination now with single agent, and these animals actually live far longer than the case. And then we went to the cyclin E amplified tumors. These are the tumors I've told you are notoriously difficult to treat. They're poor responders. They have poor prognosis. And what we found there is that the combination also had a dramatic effect on the ability to thwart the growth of these tumors, and these animals are still alive compared to the litter mates months later. So that led to the CAPRI study that just opened at Penn and Hopkins about a month ago, where we're looking at, this is a phase 1b/2 study, first PARP ATR inhibitor combination in ovarian cancer. We're looking at patients with recurrent ovarian disease, those that are resistant to platinum or still sensitive, and looking at the effect of this combination to actually not only give us responses, but hopefully really impact overall survival. And so this is a positive conclusion that led to the clinic that we're hopeful will be available to a lot of patients. Again, it's Hopkins, it's Penn, so it's sort of in between. You can send your patients there if you need to for clinical trials. And in summary, just sort of kind of restate what I've already told you about. This application is a notoriously difficult one, not only in ovarian cancer, but across many tumor types. We've identified this axis of ATR check 1 that may be a susceptibility or vulnerability of these tumors that we can now try to address with available compounds. And we've moved that into the clinic. Again, this is an open trial. We're waiting to hear about a SPORE grant that will help support some of the translational endpoints from that study. And being a pathologist, I want to just acknowledge pathologically everyone in the lab that has helped in some of this work over the years, in particular my colleagues, my new colleagues at Penn, my old colleagues at Harvard, and others that I've had the pleasure of working with. Thanks for your time. Glad to take any questions.
I have a couple of questions.
Yeah.
Well, thank you very much. It was really illuminating. The P53 gene, is it specific to ovarian cancer? Do you, because it's you find it in all ovarian cancer, but do you find it also in other tumors?
Sure. I mean, it's probably still considered the most mutated gene across cancer.
Oh, okay.
I think that what matters, or what may be different, is when do you have the mutation occur, right? So in colon cancer, it's also mutated, but it tends to occur a bit later, right? And I think it's that the loss of PP3 and checkpoint control and DNA repair later is perhaps why colon cancers are sort of by definition very mutated in other genes. They have to acquire their advantages by mutating specific genes because they still have intact DNA repair, right? Ovarian cancer, you lose DNA repair, you lose PP3 early, you lose BRCA early, you have no genome integrity control. You can get all your advantages through copy number alterations, which are a consequence of losing DNA repair. You don't have to really mutate all that much. You gain, we think you gain it by this copy number mechanism. The question is, how do you drug copy number, right? And cyclin is one example of how we're trying to do that.
I have another question.
Oh, go ahead.
So, no, go ahead. Go ahead.
I was interested, so I don't know if you've spent any time down here at the Cancer Center while you're here.
No, I'd love to.
Because there's a woman over there who's doing work in triple negative breast cancer with the liquid biopsy.
Yeah.
And so I just thought you mentioned that specifically as a
Well, triple negative is a great example because the Cancer Genome Atlas data on triple negative, one of the things that they reported in that paper is that it is amazing how similar triple negative breast cancer looks to high-grade serous ovarian cancer.
Yeah.
That if you actually cluster them without supervising it, they would cluster together.
Yeah.
Right? They're that similar. Triple negative has a very high frequency of PP2 mutations, not quite as high as high-grade serous, but in the 80s.
Yeah.
BRCA mutations are very common in that subpopulation. So we oftentimes think when we find something in one, we should look in the other because there's a chance that there's going to be some overlap in sort of the underlying biology.
We also, as a state, according to one study, have the highest prevalence of triple negative breast cancer in the country.
Wow.
In Delaware, for some reason.
It's amazing.
And we only trailed DC.
Wow. Well, that's why I think it'd be great. I mean, I'd love to talk to folks at the Cancer Center,
Yeah.
but I think the opportunities, are diverse and many for us to be able to collaborate across a lot of these things. There's the tissue, there's the liquid biopsy, there's even these studies with these companies, the falloposcope. I mean, there's an opportunity to really bring a lot and work together. As it is, it's impossible because the frequency of these tumors is usually not that high that any one institution can accrue all the patients. So you really do depend on collaborative institutions to really help with these things.
I'd be happy to connect you to those folks.
Yeah, thank you. Thank you. Yep.
So the CCNA gene, is it something that you measure when you get or that you I guess measure when you get the ovary or biopsy when someone has breast cancer?
You mean like whether routinely measured?
Yes,
So not
adjust the therapy, so to chemotherapy.
No, not yet, right? So we appreciate that cyclin amplified tumors are bad, but what's great about it is that the gene itself is the biomarker. Right, so we did some studies some time ago where we looked at FISH, which is the gold standard for looking at copy number, right, and next-gen sequencing in a cohort of patients that had or didn't have the amplification, and both results gave us the exact same patients identified, right? So that means that if you have a therapy like this CAPRI study or the AKT inhibitor combination, the way you find your patients is simply by doing next-gen sequencing or FISH for cyclin E. That's your biomarker. That's your companion diagnostic. Right, it's not any more complicated than that. And we've seen and we've done some work across other tumor types which have the cyclin E amplification, and we see the same dependence. So we think that there's sort of a common biology that is beyond what we see just in ovarian cancer. But I think what we're seeing in ovarian may inform opportunities in others. Stomach cancer is one in particular that has cyclin amplifications pretty high. Another cancer that we don't have particularly great therapy for, where these combinations in the clinic might be a good opportunity for those types of patients. And all you have to do is measure cyclin E levels, nothing else.
Right. And so do you do it? I mean,
we've been doing it, right, so we're doing it for this study, the CAPRI study, we're doing it sort of retrospective, because our therapy seems to be good for BRCA mutated, PARP inhibitor resistant, and cyclin amplified, so it seems to cut across a lot of tumor types, which is kind of unusual, but I think it's because that ATR axis is really important across many of them. But what we're going to be doing is collecting biopsies. That's part of the trial. And in those, we'll be measuring the levels to see patients who had the amplification respond or not, right? To see whether, in fact, we were right about that. So we're going to be looking at that. But right now, it's not routine practice. So I think people are appreciating more and more that it's particularly bad cancer. The question is, what can we do about it? We could find them. We just have to figure out a combination of treatment. Any other questions?
What was your favorite hangout while you were at Rutgers?
Oh, probably the grease trucks in the middle of the night, on the college Ave.
Yeah.
I used to go study there late at night because the library would close, and so this was at school when you were up studying late. And what was it called? The Fat Cat.
Yeah.
Oh, that was
Brutal.
Yeah. Well, you need a Lipitor with it, but it was really good.
So I'm sorry, I have just the last question. I'm thinking about. So do you think the future is to screen every woman because it's such a bad cancer, between, I don't know, 40 and 60 or 40 and 70?
When screening, I don't know exactly what you mean, but if you talk to Mary Claire King, she would tell you that she's a proponent of having women screened genetically for mutations in BRCA, right? So for instance, now, really from work out of Botel and Genomalis and others, right? If you get diagnosed with ovarian cancer, right, whether you have a family history or not, you should get sequenced. Because it turns out that we used to think the hereditary piece of the pie was only 5 or 10%. It turns out it's closer to 20, 25%. And in fact, it's not always in women who had a family history.
Yeah.
And so what are the implications? Well, one, if you know you have a BRCA mutation, you can get into programs that offer surveillance. There's PARP inhibitors, and then obviously there's genetic counseling that can impact the family. So it's changed now with the evolution of PARP inhibitors. I mean, we have 3 PARP inhibitors approved in the last couple of years, and we're still learning more and more about them. So I think it's changing quite a bit, but BRCA-ness is bigger than we used to think. Right? The thought is that half of these tumors have some defect. Might be a path.
Thank you.